Bom dia a todos. Hoje a palestra da disciplina Seminários de Zoologia, do curso de pós-graduação em Zoologia do Museu Nacional e do curso de graduação em Ciências Biológicas, ambos da UFRJ, será sobre a importância da taxonomia e as consequências de identificações incorretas de espécies. Será ministrada por uma das maiores especialistas em taxonomia de poliquetas, a doutora Pat Hutchings, do Museu Australiano em Sydney, Austrália. It's a great honor to have Dr. Pat Hutchings here with us to talk about the importance of taxonomy and the consequences of misidentifications. Thank you very much, Dr. Pat, for accepting our invitation, even with such a big time difference. Dr. Hutchings is a senior fellow of marine invertebrates in the Australian Museum in Sydney, Australia, where she has carried an outstanding career of research and also supervising several PhD students and postdoctoral researchers from various countries. Along her career, Dr. Hutchings has made remarkable contributions to systematic and ecology of polychaetes, to bioerosion and anthropogenic impacts in coral reefs, and to wetland ecology and coastal zone management through studies availing more than 300 publications, which includes books such as The Great Barrier Reef, Biology, Environment, and Management. All these outstanding contributions to marine research were recognized by the Excellence Award Jubilee Medal that she received in 2010 from the Australian Marine Science Association. She is current editor-in-chief of the Marine Pollution Bulletin and editor for Annelida in Zootaxa, and leading expert in polychaete systematics, phylogeny, and ecology. So without further ado, we see you, Dr. Pat Hutchings. Thank you very much.
Thank you very much, Joanna. And Joanna has actually spent some time here in Sydney with me. So she's actually going to be, she's a co-author of some of the papers that I'm going to be referring to. So as Joanna has already said, the talk is why is taxonomy so important and the consequences of misidentifications. What is the problem is that polychaetes are a diverse and abundant group with something like 20,000 described species, and more species are being described all the time. And increasingly, we're finding suites of cryptic species as molecular work is undertaken, and also as we're improving the quality of our morphological descriptions. Polychaetes are found in a very wide variety of marine and estuarine environments, but they tend to dominate benthic communities, both in terms of numbers of individuals as well as their diversity. So it's a really important group to correctly identify because they actually play a very critical role in benthic ecosystems. So what are the knowledge gaps in polychaetes? While there's a large numbers described from intertidal to shallow subtidal areas, the deep sea is poorly known in contrast. Historically, most polychaete workers were based in northern Europe, and these workers obtained specimens from major expeditions, such as the Challenger, which went around the world, Siboga, which was mainly South Indonesia, and various German expeditions, including some of those to South America. The material was collected from areas that previously where the fauna was poorly known, but all this material is currently deposited in European museums, and sometimes it's not all that easy to find. And also, museums like the one in Hamburg were bombed during the Second World War, and so types were lost. Later, there were United States expeditions, and that material was also deposited in US institutions. Just as an example, there was no polychaete worker in an Australian museum until 1970 when I arrived, and we now have a very active group of workers, and you can check out on the ALA, the Atlas of Living Australia, polychaetes that have been described from Australia. South America is very poorly known, or it was until a few decades ago. And now you have an active group of workers. And in part that was due to an international polychaete conference that was held in Curitiba in the late 1990s, early 2000, by Paula Launa, who I think gave you one of your talks at the beginning of this series. But Southeast Asia and India are still very poorly known. Polychaetes from deeper waters tend to be very poorly known, and as you get into the abyss, very poorly known. We're currently revising a manuscript that's looking at the polychaetes from the abyss off the east coast of Australia, and we've not been able to put names on most of them. So when you've got such knowledge gaps in polychaetes, it's really difficult for benthic ecologists to correctly identify their species. And there is a tendency, it's much easier to put a cosmopolitan name on it because of the time that it actually takes to correctly identify polychaetes to species. It's not saying you can't do it, but it does take time and effort. I can see Joanna nodding there because, that's really true. Now, the consequences of this knowledge gap is that people in many countries use inappropriate literature to identify their fauna. They often rely on Fauvel, which was published in 1923, that was the errants, and 1927, which was the sedentaries. And or else they use Day, 1967, a monograph of the polychaeta of Southern Africa. Both are old, and on reflection, one is almost 100 years old and the other is 50. For instance, with Fauvel, while it was very comprehensive at that time, we have moved on. Now, for instance, I've just had Nicolas Levesque working in my office. Unfortunately, he had to go back to France because of COVID. And in one of the papers that we've just published, we described 9 new species of trichobranchids from France. And the only terribility species which was listed by Fauvel for this family doesn't actually occur in France. It's actually restricted to Norway. So, and that's just the beginning. I mean, it's just, I don't want to in any way denigrate Fauvel's work, or if for that matter Day, but we have moved on. Science taxonomy is an evolving discipline. And people who say, oh, well, you could just identify polychaetes and, it's just stamp collecting. It's not. There's a lot of work to be done out there. Now, I'm going to give you a case example of Malphiza sanguinea, and Joanna knows all about this too. It was originally described by the Reverend Montague back in 1813, and it was actually described as not as a Nereus, not as Malphiza. And I apologise, Montague 1813 should be in brackets. It was originally described from Devon in the United Kingdom. That's down in the southwest of England. And it was found intertidally in rock crevices from a place called Wembury. It's now a national park. It was very poorly described and no type was deposited. But, this is 1813. And, you can see there at the bottom, that is the original plate. And, it's obviously unicid, but not much. You can't say much more than that. But that was the standard of descriptions in those days. I mean, let's not be too critical of the Reverend Montague. And also no type was deposited. But that was typical. It wasn't, that wasn't the way it was done in those days. So this poorly described, or this very sort of poorly described species, it allowed this species to be reported from around the world in a wide variety of habitats with a variety of temperature regimes. People didn't even consider that this species occurred in very different habitats. Some of them were described in seagrass beds, in muddy substrates, as well as in intertidal crevices. And so also And there were some cases where species of Malphiza were described, but then they came along and said, no, they're all Sanguinea. It's really a widespread species. And it's only recently that we've realized that polychaete species tend to have very restricted distributions unless proven otherwise. And I'll come on to that in a minute. So what needs to occur? And I mean, when I first got to Australia, we had Sanguinea that was the species that was present in Australia. And, it just didn't seem right. The species in Australia that was found in seagrass beds, in warm water. I mean, the temperature in southwestern England is not exactly warm. So what needs to occur? First of all, we need to re-describe the species using modern techniques like SEM and hopefully molecular. But first of all, we had to find a specimen. So back in the late 1990s, I searched the collections in the Natural History Museum in London. And I found some material that had been collected from close to the type locality. It was, about 20 miles away. And it was collected in the 1850s from an estuary that was very, was not a port, so it couldn't possibly have been introduced as an invasive species. It hadn't been substantially modified in the 1850s. So I designated one of these as a neotype, and I described it in detail together with a student of Peter Olive's, who was at that stage working on the reproductive biology of this species. And we subsequently obtained fresh material, which we sequenced, and Joanna was involved in that project. So now we have a fully described species. We now know what Malphiza sanguinea is, but based both on morphology and molecular. And we've now found that it's restricted to the English Channel and also to Arcachon Bay in France. So once you've worked out you can define your species, you can then check all the records of Sanguinea from other parts of the world and describe them either as a new species, or you could say, okay, we really do have Sanguinea in South Africa or India or whatever. And it's almost like a detective story. So, it's a good detective story. So when you look at all those records of Sanguinea from around the world, you can only really confirm whether they're Sanguinea or not if somebody has actually deposited some material. For example, Wild Day in his book in 1967, which is widely used because it's got some nice illustrations in and everything, and it's a compact book. We couldn't even find any specimens on which he had based his description of Sanguinea from South Africa. And it's obvious that it was a composite description of Malphizas in general. So we couldn't actually confirm his identification. Whereas a group, some scientists in South Africa found some fresh material, and they described it as a new species. Subsequently, and Joanna was involved in this too, it was revealed to be a junior synonym of a previously described species, Malphiza hemosoma, which has now been reinstated as a valid species, which had previously been synonymized with Sanguinea. But the name Sanguinea is still present in the literature, and it's really depressing. It's been repeated in the checklist of South African polychaetes, which is, I think, still in press. Very depressing, but we'll get there somehow. Also, it's been recorded from India by Fauvel in 1953. It was probably his last publication, but there were no vouchers deposited. So recently I received some Malphizas from India, from someone who was doing her PhD on Malphiza, and we've just described it as a new species, and we could find no evidence that Sanguinea actually occurs in India. Similarly, records of Sanguinea from China could not be substantiated. And I had a student, Yubin Liu, who came and worked with me for a year. And he came and said, I want to do the reproductive biology of Sanguinea from China. And I said, well, first of all, you should check whether it actually is Sanguinea. And he'd collected his, he'd bought his, he wasn't actually a field biologist. He bought his worms from a local bait collector. And we managed to find that there were 5 species described, 5 new species amongst that material. And again, no record, no evidence that there's Sanguinea in China. I mean, perhaps some, you can never say categorically it's not present, but we found no evidence that it's present. Also, records from the Caribbean have also been shown to represent new species or species that were previously synonymized with Sanguinea, and those species have now been resurrected. Now, this is Nicholas. Back in his lab in Arcachon, and he was interested in the Malphizers that were present in Arcachon Bay, and where it had always been where Sanguinea was reported. Certainly Sanguinea occurs there, but there is Victoriae, which is found in soft sediments, immediately a habitat that's not the same as Sanguinea. Now, why is this animal? You can see there the length of it. It's highly prized as baitworm, and it's shipped all around Europe and sold for, a good length of worm like that costs quite a lot of money, and the situation in Arcachon is confused because there is actually Sanguinea there, but it occurs in a different habitat to where Victoriae is. And we'll tell a later story in a minute about another species that's in Arcachon. So why is all this problem with Malphizer? And remember that around the world it's used widely as fish bait, and there's a vast industry associated with people buying. I mean, recreational fishing is a very lucrative area. I mean, in Australia, you go to the bait shops and you can buy a packet of worms to go fishing, and it's worth a lot of money. So why is there all this problem? Is that basically all species of Malphizer look fairly similar. You need to examine in detail the type and distribution of chaetae, the shape of parapodia, and the distribution of branchiae, for example. But you must consider the habitat. And increasingly, we've now described probably about 12 new species of the Sanguinea complex. And they've all got, is that they've all got very discrete distributions. But as Joanna and I found, even in the Hawkesbury around Sydney, you can have more than one species present in the same sort of region. While the two occur on the same beach, perhaps one's more associated with seagrass beds, whereas the other one's in a much muddier habitat. But you can't distinguish them in the field. Recently we had a bit of another detective story. Nicholas and I went to the Polychaete conference in Long Beach last in August 2019, and we met a Japanese guy there, and he said, I've got a species that I've just described from Japan that looks very similar to what you've described as Victoriae in Arcachon. And so he said, can I send you some material? So we compared the sequences, and guess what? The species in Japan is actually the same as Victoriae occurring in Arcachon. So how did a Japanese species get into Arcachon? Well, you go back. Arcachon is a very important oyster growing area in France. And I mean on a major commercial basis. Back in the late 1960s, early 70s, the entire oyster industry in Arcachon collapsed due to a parasite or something. I'm not quite sure. And so they had to restore those oyster beds, and they imported live oysters from Japan. And it's our suggestion is that while they brought in these mats of oysters, in amongst them were either juvenile Malphizer from Japan, or as perhaps even slightly, or just egg sacs, because some of these things actually have egg sacs, and where the eggs develop and then get shed. So we've actually therefore got good evidence that those oysters, that Japanese species is actually hitchhiked with live oysters into France. Now, actually, we don't actually know, even more complicated than this, because from China, live bait of Malphizer is shipped to Southeast Asia, including Japan. So we don't actually know whether that species that was described in Japan is originally a Chinese species. We haven't quite solved that problem yet. So, it's a story behind a lot of these species. Now, this is a species that Joanna and I described, and it just shows you this type. You remember that photo, that plate I showed you about 4 slides ago, as to how the original Montague described Sanguinea. These are the sorts of images we now always have when we describe a new species. The front end, you can see there the jaws, and if you look, and also we look at the structure of the parapodia and the SEMs of the Kiti. So a lot more detail is now used. And on the right here, we've got molecular data, and you can see there where Sanguinea fits in and where bifurcata sits in. So what are the consequences of this misidentification? Is that Sanguinea is still the occurrence of Malphizer Sanguinea is still present in the literature in places way away from, a long way away from Devon in the UK. And even some of the records on GenBank, in some cases, they're not even a species of Malphizer. So I'm afraid some taxonomists just repeat lists of records and distributions of species without actually checking all those records. As I've already said before, species have restricted distributions unless proven, like being transported by agricultural practices or perhaps by wholesaling. So there are some species that have wide distributions, but they're artificial. They're not natural. Apart from, there are a few species of polychaetes that are pelagic, and that I'm happy for them to be they're transported by ocean currents. A few years ago, I worked on some terebellids down in the off the coast of southern Brazil with Miriam Schuller, and we had a species of terebellides, and it was separated by about 2,000 kilometers. But it was in deep water, and when we looked at the ocean currents, there was good evidence that those larvae could be transported around in that basin, and also the molecular data showed that it was the same species. So we taxonomists can be guilty of spreading these widespread species, such as Malphizer sanguinea. But what are the consequences of these misidentifications? It means, ecologists undertaking benthic surveys need to identify their fauna. They've often got limited time and limited funds, so they use the available literature, or they just use lists from previous ecological surveys. And often there is limited communication between ecologists and taxonomists. And a lot of ecologists are not very good at depositing vouchers in the local museum. So just using Sanguinea as an example, and there's plenty of others, we have records from around the world. But we know that even in the same geographical area, 3 species of Malphizer can be present. For instance, in Moreton Bay, which is north in Queensland, just north of, on the east coast of Australia, at the southern end of the Great Barrier Reef, Marphysa mullawa in seagrass beds is harvested commercially, but it's not the only species that is present. Another we've described from the mudflats, and another from the mangroves, all say within 100 meters of each other, and all belonging to the Sanguinea complex. And as I've just said, we've just described 5 new species from China. So Marphysa has been used as a biological model in biochemistry for biological and physiological studies, including reproductive studies, rearing of juveniles, because it's important as fish bait. People have been trying to rear Marphysa commercially so they can sell them. And I mean, a kilo of Marphysa, bloodworms in Australia, is worth more than a kilo of fillet steak. There's genetic studies, there's suggestion of microplastics, etc. But who the hell knows what species are involved? So no vouchers deposited. So basically all that data is worthless. Because even if they're all Marphysas, each species has its own characteristics. So, people are wasting their time, and people, that data is in the literature, but it's not worth it. So as I said, we've already explained that we just, often taxonomists just repeat their records and things like this. So what is the value of all those papers of Marphysa? Basically zero. Who knows what they were working on? Whose fault is this? Some of these papers have been published in international journals. Says something about the peer-reviewed papers and also editors. I know when I review ecological papers, when I question the identification of material listed, rarely is any notice taken by the editors or authors. We're all responsible for some extent. So this is really depressing. In times of limited funding, it means that much of published data doesn't actually have any value. The correct identification would actually create useful knowledge, increasing our knowledge re the biology, ecology, the reproductive biology and physiology of this work. Instead, we have papers of no value and again, spreading the myth of so-called widespread species. It's a waste of valuable resources. So why does it matter? What is actually a name? It's like, I think in Brazil you have identity cards. A name basically identifies what species it is. It's like, and when you go, for instance, when you go to the bank, you need to provide evidence in order to establish a bank account. You need to show your driver's license, your passport or something to get a driver's license or to get a passport. Your name attaches a whole load of criteria to you. I mean, like when I try and go to Brazil, I have to provide, I have to show my passport. They want to know who I am. So each species has its own unique characters, not only in terms of morphology, but also in terms of when it breeds, how it breeds and feeds. It has specific habitat requirements, physiology. And if you continue to misidentify the species, but continue to use the name, you are still assuming still has all the same characters as the species name you have stolen. So for example, if you use Sanguinea, now back in England, it breeds at 19 degrees. When we know that mullawa, which was originally identified as Sanguinea, breeds at 29 degrees and breeds over an extended period, whereas back in England, it only has a very short breeding cycle. You are losing valuable data. And just because and all species within a genus don't necessarily even do the same things. I mean, they may all have the same feeding habits, but probably not. So that, each species has its own unique characteristics. So how do we resolve this issue? We need to improve the dialogue between taxonomists and ecologists and biochemists. And I really encourage students in Sydney to come and see me when they're doing their ecological work, and I'm happy to try and help them. We taxonomists need to become more vocal, and we also need to be better recognised by the scientific community. There are people out there in the scientific community who say, oh, well, we know all our animals, we know the fauna, you're just stamp collectors. As I've said, taxonomy is a constantly evolving top science. We also need to stress the importance of vouchers and that they're deposited in a museum and available to be found. And increasingly, as museum collections are data-based, it's not that difficult. Some museums might, I think with COVID, we're going to have problems that some museums are going to have major financial problems, but we need to ensure that the collection managers understand why vouchers need to be deposited, and the ecologists need to work with those collection managers, because collection managers don't just want a whole load of whole load of specimens. They want to have proper habitat data, location data, lats and longs and everything. So they need to have the material given to them with all the necessary information. I know that our collection manager, Steve, will not accept material unless it says exactly where it was collected, when, how, sediment type and all that, so that it can all be put into our database, into our EMU. We need to make editors more aware, so that, when you, later on in your career, you review papers, you need to check that materials has been deposited. Are there authorities for the species cited? But I do despair. We can become much more vocal, not just within the taxonomic community. We need to be more proactive within the scientific community, and we need to develop interactive keys, monographs. We need to make it easier for people to actually identify their animals. I'm currently working with some people here in Australia to update an interactive key to polychaete families, and also to update interactive keys to identify species within particular families. And these, as they are produced, they will all go onto the web and be freely available. We also need to make sure, and I think Joanna is playing a good role here, that students at universities become far more aware of the value and importance of taxonomic studies, so that when they go off to become ecologists or invasive species biologists, they understand why it's so important to have the correct name. We need, but perhaps scientists in other disciplines like some of the physiology or genetics is perhaps far more difficult to reach. I can remember very early on in my career in Sydney, somebody phoned me up and said, "Oh, I've just done some genetics. I've just on the worms, some worms from Carril Bay. Can I have the names?" I said, "Well, excuse me." I said, "There's about 150 species described from Carril." "Oh, it was long and narrow and sort of red, and I said, "Well, that doesn't help me at all. Can you bring me a specimen?" "Oh no, we've ground them all up. We've done it." Again, so useless. And I suppose a point that I often make is that how can we manage our marine ecosystems if we don't actually know what's there? How do we, say we develop establish a marine park and we establish zoning plans, how do we know whether that zoning plan is actually working if we don't actually know what's there and how it's changing? We can say, right, we've got some more fish as a result of having a sanctuary zone, but what's happening to the animals down in the benthos, the ones that are supplied at the bottom of the food chain? That's why it's so important to correctly identify your animals. I know it's it's not easy, but go and talk to your relevant taxonomists and work with them. And often it can be a mutual two-way system, because we taxonomists often need material collected by the ecologists to do our taxonomy. I think I've probably done enough. So I'm happy to take a few questions, but I am going to go to bed soon, I have to confess.
I'm on mute. Thank you very much, Pat, for the wonderful talk. I hope everybody enjoyed as much as we did. And I'll just thank to the people that are watching us on YouTube. I hope you enjoyed us. Espero que todos que estão no YouTube tenham gostado, tá? E a palestra completa vai ficar disponível no canal do PPG Zoo. Okay, I just had to stop